Hi and welcome back to our GCP-Mindset channel and all topics on clinical research. In Part 9, we'll give some conclusions on statistical tests in clinical trials. More after the intro. In conclusion, it should be noted that sample sizes facilitate an approximate prediction of the required number of subjects. In this context, it is important to consider carefully how many subjects are needed in order to, first, avoid overlooking the effect, avoiding beta errors, secondly, to avoid identifying non-existing effects, avoiding alpha errors, and thirdly, to avoid enrolling more subjects than necessary. After this summary, still the question arises why clinical drug studies are generally bigger than clinical medical device studies. While in some cases about 5,000 patients are included in big clinical drug studies, there are in general not more than 100 subjects included in studies testing the performance and safety of medical devices. This phenomenon can be explained by the different modes of action. A drug has pharmacological or immunological effects, whereas a medical device works physically. The pharmacological effect of a drug can lead to multiple systemic side effects, which means it can influence several organs like the kidney, the liver, the central nervous system, and many more. In most of the cases, these side effects occur in just very few patients. The variation of side effects is relatively high. In order to get significant results, many patients are required. In general, the physical effect of a medical device is just local, meaning it occurs where the device has been applied. Side effects such as local irritations or inflammations are relatively frequent. If the physical adverse reaction has occurred for one patient, the probability is quite high that it will also happen to the other patients. The variation is relatively low, therefore less patients are needed for proof of concept. Furthermore, in drug studies, many organ systems have to be monitored, leading to a variety of different data, such as different liver values, renal functions, enzymes, and others. The local side effects of a medical device can be determined based on few parameters. The same applies for the efficacy. While the efficacy has to be proven for each drug, for a medical device only the performance has to be demonstrated. The proof of efficacy is based on several factors, such as the dose and the duration until the effect is seen. The proof of performance is in contrast relatively simple. You can compare it to the performance of two cars going from A to B. Car A is an economic small car. Car B is a high-output SUV. Both cars show the same performance to go from A to B. Such a proof of performance would be easy to accomplish, and SUV would certainly show more power. However, the efficacy of the two cars depends on many factors such as energy consumption, speed, CO2 emission, type of gas, wind resistance, and many more. All these parameters would provide information about the efficacy, as it is also the case with the drug efficacies. The efficacy is mostly described by a variety of primary and secondary endpoints. This leads to several interactions and multiple test problems for statistical proof of efficacy, which often requires an adjustment of the alpha error and a large number of study subjects, whereas the performance of a medical device can be proven by one simple endpoint. Interactions of endpoints can thus be avoided. An alpha error of 5% can generally be applied, and the patient number required for the proof of performance is smaller. So much for today on statistical tests in clinical trials. We hope that we could give you some interesting information and look forward to see you next time.